Yeah, so you forgot the last bit of my title, which I added yesterday, because I realized that's probably actually the direction I'm going to talk about more. But yeah, so thank you very much for setting up this school. I think it's really great, and thanks for inviting me. So today I will talk about networks. We've already heard a lot about spiking neurons, and that they are basically the driving force of computation in the brain. But what really does the computation, and the problem that we're really interested in, is how does the brain take in sensory inputs on the one hand and processes them to turn out ultimately intelligent behavior. And we often come up with abstractions as computational neuroscientists, where we really abstract away a lot of this complexity by saying we're looking to find a functional description of what's going on, like a function f, with some parameters theta, that takes in a stimulus in an abstract form and outputs an output. So I think we've already seen this yesterday in the introduction session. That's a little bit very restrictive view of what the brain does. We'll usually also have to take into account that the system itself, so the network itself, the animal itself, can have an internal state, it can have internal expectations, and it can have integrated some notion of context, for instance, that should also inform this function. So this is already a vast abstraction for what's actually going on, because of course underlying stimuli are hidden causes in the world, and the outputs, they're usually motor actions that allow us to interact with the world. And so these are actions that will change the world or change our position in the world. And for today, we're not going to go into this, but we really focus on this mapping here in the middle, right? So a vast abstraction we're using, and we should keep this in mind. Now, of course, we know that this function is implemented by neural networks, right? So neural networks, basically what the brain consists of. And these networks have a lot of structure, and you've already heard a lot about that previously. But for instance, we know the different brain areas. So for instance, sensory areas, motor areas, and some associative areas in the middle. So that's something that's stereotypical or preserved across individuals of one species, for instance. And if you now zoom in to the circuit level, again, you have a lot of structure, right? So you have different neuron types, they connect to each other in very specific ways. And presumably this is all important, and it's not entirely clear how this works. And then if you zoom in even further, even to the smaller details, to single neurons, again, you have a lot of complexity that network modelers like myself often like to ignore. But Hermann will, for instance, talk about a lot of the neuronal complexity that you have just because of different morphologies that neurons can have, that allows them to do more complex things that you would often think a neuron can do. Right. So here is an example of a neuron model. In this case, this is work from the Gerstner lab. And it's what's called a generalized integrated fire neuron model. And today, I think also in Sasha's talk and in the afternoon tutorial, you will learn more about integrated fire neurons. But they're basically the simplest type of neuron models around. Network modelers like to use them because they're efficient to simulate. But they're also pretty good, often at least phenomenologically, in capturing data. So and here I'm showing you the response of a real neuron, here per layer 5 pyramidal cell recorded in the Markram lab, overlaid by the model in red, so black is the real neuron. And this is a response to this noisy injected current that you see here at the top. And you can see the model does, and this is on held out data, I should say, the model does a really good job at capturing here the subthreshold dynamics and also capturing the spike times. And these are multiple trials, and you can see this as even a stochastic process. So that's just to say that for these type of single neuron models, these effective neuron models, we have a pretty good description of the input-output mapping of individual neurons. So a pretty good understanding, you could claim, of what's going on. But if you go to the circuit level and the network level, where ultimately the function arises, this understanding is not that good yet. And that's where we want to get. So the reason why we lack this level of understanding is really that the link between the connectivity and the function that the network has is not very well understood, neither theoretically nor in simulations. And somehow you need to have this transition, right, between a network that's naive, in a sense, which doesn't have a specific structure in its connectivity, to one that's not naive, that has a specific structure in its connectivity that gives it this function. So not every random network model out of the infinite, literally infinite combinatorial space of possible networks will give you a useful function. Most of them won't give you any function if you want. And in my group, we approach this really through a combination of top-down and bottom-up modeling, where the bottom-up end is really that we put in biological constraints, such as spiking neuron models that are realistic, or plasticity models similar to the ones you've heard about this morning, and that we inform these models with specific connectivity motifs or other kind of connectivity constraints that we know exist in neurobiology. But at the other end, we also realized that just now putting plasticity in such a network model is usually not enough. So just having data-driven plasticity models such as SCDP that have been fitted to some data and putting them in the network model usually doesn't automatically makes your network intelligent. So you can't then teach it languages and go to the cinema with it and have fun with it. So it's we're still far away from this. Basically, these models, they're not necessarily smart. Usually things synchronize and blow up. So Hebbian plasticity alone, or these type of plasticity models alone, they're not enough. So we kind of realized, and that's not only us, it's the field as a whole, and many of us have realized that you want to also have these top-down constraints, functional constraints, because ultimately what a neural network does is it processes information. So you want a model that is also capable of doing this. And somehow you want to be in this sweet spot between biological constraints and the functional constraints, where the model does something and it does so with biologically plausible components, so you can actually learn something about the underlying biology. So that's the goal, and it's with all modeling, the goal is to identify the general principles from this, to really make predictions that can then be falsified or verified for validation and understanding. And it's an iterative process with the experimental colleagues. So to get these functional constraints in the type of modeling we're doing, we've taken over the years more and more a deep learning-centered approach to the problem. So that's really where the machine learning comes in. So in this case, the machine learning actually serves as an inspiration for the biological modeling. So the arrow goes the other way around, which is why I added this to the title. And what the machine learning folks really have figured out is how to build big network models that do something. Right? So they don't necessarily focus on these network models being realistic and brain-like, but they've definitely figured out how to get network models to do something. For instance, speech to text or translation or object recognition and things like this. Nowadays we have self-driving cars, so it's really amazing what deep neural networks have given us there. And underlying are neural network models, artificial neural network models, that also need to have a specific structure, right? And the way these networks get this structure is through a learning algorithm that ingests enormous amounts of data. And that's really part of the inspiration we're also currently taking for this biological modeling. And so there are three ingredients really that you can break this approach down to. And one is really concerned with the architecture. So how many of these areas do I have? What are the types of neurons that are in there? How do I connect them on average? How do I initialize the weights? Then usually what you want is you want to somehow characterize the function you want this network to do in a way that you can phrase in an objective function. So that's the function that measures how well am I doing at the problem I want to solve. And if this has a high value, then I'm doing really badly. If it has a low value, then I'm doing well, right? And then the machine learning trick is really that you basically can derive a learning rule from this objective function by simply taking the gradient. So this is a very high dimensional parameter space, but because everything is initialized in a way where it's differentiable, you can literally take the gradient and follow the negative gradient, so you find a minimum in this loss function. And then with a bunch of other secret sauce ingredients, this minimum is a good one, which generalizes well, so you have a network model in the end that solves your problem. And so the three components are really the architecture, the learning rules, and the objective function. And we can think of them separately, and we can think about all of these difficult problems separately, and break apart a very difficult problem into three slightly less difficult problems, which is the usefulness of this abstraction. So in artificial neural networks, as I already said, is a training algorithm, and I'm not going to explain to you backprop, but just to repeat this, the idea is you have an architecture to begin with then you have a lot of data. So you need a data set, a training set. And then you usually, in the supervised learning setting, you have a desired output, and the network initially will not match that desired output at all because you haven't adjusted all these parameters the network has. So there will be an error, and the objective usually is to minimize this error. And then you use an algorithm that computes the gradient, which it does by introducing essentially feedback that goes to these different areas or the different layers in your network and tells you how you should update the weights to decrease this error. And you do this iteratively, and then ultimately, what ideally happens is they minimize the error and the network generalizes to new data. So it's arguably something that might be very different to how the brain solves this problem, but what was really reassuring to see roughly 6-7 years ago was work from the DiCarlo lab and also from by Nico Krieger's cohort, where people figured out that if they take a deep neural network that has been trained on a visual recognition task and they compare the activity in these networks to monkeys that had been trained on the same task, and they compare the activity between the layers in the deep neural network and monkey brain activity in the ventral stream, they find that they share representational similarity. And that's what's shown here. So this is basically a neuron in black and monkey IT. And on the x-axis, these are different images that are shown to the monkey. And in red, you have, in this case, it's a cell basically in a deep neural network that has been trained on the same problem. And there's one minor linear alignment step in between. But otherwise, basically, you see there's a striking similarity. Right? And here, what these matrices show, what you should see is there's a similar structure. These are called representational similarity matrices. So each pixel in these matrices basically corresponds to an angle and of two representation vectors corresponding to two image inputs to, in this case, a human, a monkey, or an artificial neural network. So it's like a scalar product. And you can see that this geometry seems to be preserved, even though this has not been fitted to this, right? So they were just solving the same problem. So this is kind of reassuring that maybe this deep learning holds some good ideas for us to understand what's going on. So that's why there has been some recent excitement about these deep neural networks, but here recent really has to be seen in the greater context of 30 years. So nobody else than Francis Crick at the time noted that the remarkable properties of some recent computer algorithms for neural networks seem to promise a fresh approach to understanding the computational properties of the brain. Unfortunately, however, most of these neural networks are unrealistic in important respects. So, and Crick was absolutely right. So the way we train these networks for starters is very different to how the brains learn. So the learning algorithm, so that's here the objective, right? So supervised learning is not what the brain does. Then the learning algorithm uses implausible feedback. It has to do with weight symmetry. And Ozra already showed earlier this paper on backpropagation in the brain. So I recommend reading this. So there's this whole debate how the brain, or whether the brain uses backpropagation in some form to learn. So the learning algorithm uses implausible feedback at the time. And then also importantly, and that's what we are going to talk about now, is there are no realistic spiking neurons in these models typically, no cell types, no circuit motifs, if you want. And now, more recently, there's been, again, excitement about this because people started figuring out how to train networks with more realistic spiking neurons. That's something I will tell you about now. And how to overcome these problems with implausible feedback. There has been a flurry of papers over the recent years. And I will also, in the end, talk a little bit about some recent work in the lab where we're now getting at this question. Good. So this is really the focus of current research in my group. So today, really the main part, part one, is show you how do we train spiking neural networks using these ideas from machine learning, and then some theory on learning without actually labels. Right, so for part one, the idea is we want to take backpropagation or the type of learning algorithms, put them into spiking networks and get spiking networks that do something. On the one hand, to build plausible network models that do something, but also, and we'll do this both simultaneously, although if I had more time, I would do it separately, we will simultaneously also look into these algorithms, into how we can shape them so that they become plausible in terms of what a real neuron could implement also as plasticity. And that should hopefully tie up nicely with the plasticity models you've seen earlier today. Right, so suppose you have now specified a network architecture, and you will all play with some networks later on. We have to talk about inputs. So if you're interested in spiking neural networks, so there were the three things, remember? Architecture, input data, learning algorithm, and the objective. And so inputs should be spiking too, ideally. So we would need large data sets. And that's just to give you some examples how these different sensory modalities could look like. So for touch, for instance, here you see spike trains. So this is time, different neurons, and then different trials within these blocks. And the different colors correspond to different textures. And that's a primate basically with a finger moving over these textures. And you see there's a lot of spatiotemporal structure in these textures. So this is how, presumably also in your brain, when you touch the surface, how this sensation is encoded and goes into your sensory cortex. So there's a lot of time encoding here, obviously. So for vision, that's actually similar. So here's an example recording from a ganglion cell in a salamander retina that's looking at another salamander. So going along this axis here in this salamander picture, you see the response that corresponds to this. So it's an off cell that likes the dark pieces of the image. You can see nicely this modulation. And here you have a reconstruction of this encoding, of this visual encoding, where you use either relative latency to the first spike or a number of spikes. And you can already see that actually the latency, so the time of that spike encodes a lot about the stimulus. So this is discarding all the other spikes, and the rate also does. But so this is already probably one important clue for us that maybe the brain uses this temporal dimension in some places where it's useful to save spikes, for instance, and thereby saving energy. So ideally, we want to come up with learning algorithms that can do that, right? So that can live up to that goal of using this timing encoding in an efficient way. Finally, that's an artificial data set that we actually created, because for this approach you need large data sets, and often they're not available in recordings because you need thousands, 10,000s of inputs. So we built, in this case, Benjamin Kramer built a cochlea model and harassed all of his friends to record basically a speech data set with spoken digits. And they're fed into this model. And this is somewhat similar, so we hope, to the spikes that your cochlear nucleus would spit out into your auditory cortex when somebody utters the word 6 in this case. Right. Good. So for pedagogic reasons, we're going to resort to a much simpler model for this talk, but this would usually be the types of inputs that you'd be working with. What we want is really the simplest thing we can possibly imagine right now to understand how we do this. So we'll have different examples which will correspond to spike trains, so time, different neurons. And what we want is a network, a spiking neural network that converts input spike trains into some output spike trains. And we'll start with supervised also, so we'll have the target basically here on the right-hand side. And now this is already a network with a hidden layer, as computer scientists call it. Let's start even simpler. Let's get rid of the hidden layer. Then the derivations become much more manageable. And so now we need to talk about the output end of the network, right? Spikes come out. How do we train this network? We need to define an objective function. And for the task that I've just described, what you want to do is we need to define a loss function that, if the task is to learn a specific output spike train, we need to define a loss function that kind of captures this. And by the way, so I see people taking pictures. I'm happy to share the slides later if you want, but so we can send it around over whatever. Right. Good. So let's focus on this one neuron, and we want this neuron to learn a specific target spike train here in blue, but it puts out these two spikes that are wrong, right? So the output, there's an error now, so it doesn't put out the right spike train. So what's the van Rossum distance? The van Rossum distance is basically written down here. So it takes in the spike train, and that's, in this case, just the sum of Dirac deltas. That there's a Dirac delta at every spike time, basically TL. And you just take the difference, you filter this with a temporal kernel, which looks causal. What we're going to use, sorry for the online people, looks like an ePSP. We're going to use something where this is time as a kernel, looks like this, okay? So it looks like an ePSP. So you filter this here, and then you square this difference, and then you integrate over it. And you can, I think, immediately see that if they're the same, then this whole thing is 0. And if they're not the same, this is going to be positive. So that's a good loss function, and you want to minimize it to drive that learning. Good. So then gradient descent. So you take now derivatives of this loss function with respect to the parameters. The parameters in this case are weights W, and they're somewhere in behind this output spike train here, S. Right, so this is a network model. So it has an output spike train. The output spike train depends on the membrane potential. The membrane potential depends on the spikes of the neurons in the previous layer, and so forth. So it's a complicated function, but somehow it depends on W. So, and if you take the derivative, then obviously this 2 goes down here, cancels with this. You get this entire term here. Then you need to take this inner derivative, which is written out here. Convolutions are linear operators, so you just carry them through. So nothing happens. And now you have a problem. So here is the problem occurs because remember this S has these direct deltas in it, right? So now you're screwed. Good. And the problem is really that this is at the heart of our spiking models, that they have a binary output, right? So there's either a spike or no spike. There's no half spikes, like in a nicely differentiable neural network model, artificial neural network model. So if there's a slightly higher current here at this point, so this neuron crosses threshold, you have a spike. If the current is too small, there's no spike. So it's in a way, in voltage space, where this is voltage, it's like a step function, like a Heaviside function. And that at firing threshold switches. So derivative here is 0, derivative here is 0, and here it's infinite. Right? So that's an issue. Yes, there's a question. So maybe a microphone or I don't know.
Can you maybe just grab the microphone?
Yes, explain the equation.
No. You said there was a problem in the equation, just because I'm a layperson to the complex mathematics. Maybe is there a way that I can
Right, so I'm
it?
actually trying to explain it right now. So the problem in the equation is that you want to compute derivatives of this equation, and you can in this case, and you have a component in this equation where the function itself has a part that's a step function. All right, so that's because neurons spike. And now if you take a derivative of such a function, here the derivative of this function with respect to the voltage is 0, here it's infinite, and here's 0. And all of this uses chain rule. So chain rule is a basic rule that you use to evaluate these concatenations of functions, the derivative thereof. It gives a huge product of derivatives. That's what the chain rule is. So, and if you have in a huge product one number that's either 0 or infinite, the whole product is 0 or infinite. And in consequence, you can think of the whole problem as an ill-conditioned optimization problem, where now your loss landscape doesn't look like this nice thing that you can walk down a gradient, but it looks like an inverted pyramid in Egypt, where you have this kind of local structure that's just steps, right? So it's flat in most places, except where there's an edge it's like a cliff. So the gradient is 0 almost everywhere, so you can't use the gradient as a local signal to learn. And what you, of course, want to do is to find an approximation that smooths this out again and allows you to do it. And similarly, you want to smooth out this edge. And that's what people have done extensively. There are different approaches. One is to inject noise. Another one is to actually make the spikes differentiable, but then they're not really spikes anymore. So that's it's one approach, especially if you care about implementing this in hardware. It's also not very efficient. This one is a technical one, which I'm going to skip. But so the option 4 that we're going to use is what we call surrogate gradients. So this is right now our go-to solution. So we'll use another gradient that doesn't correspond really to the system, but it has these properties and it's close enough to the real problem in a way, or it's informed about the real problem that helps you solve the real problem. And the way literally we do it in this large product that I just mentioned, whenever you take the derivatives in the large product, and you encounter a Heaviside function, you just say, oh, there's a Heaviside function that's going to blow up everything. I'm just going to replace it with a sigmoid or something. But you're only doing this when computing the derivatives. You're not doing it in what we call the forward pass. So you still have a spiking network, still binary spikes. So it's not the true gradient anymore of the system, but it's one that has the properties that you can use to go down a surface. Right, so this solves the issue of this function, and then Again, here this is just showing the functional form of the membrane potential of a spiking neuron in what's called the spike response model. So it's an integrated form of the neuron. These are the presynaptic spikes. They're filtered by a kernel that looks like this. So to give you an EPSP, actually I think I have it, yeah, this is the kernel. And weighted by the weights, the presynaptic weights. And then the reset, spike reset is basically modeled by this part, so this index is the same here, so this takes care of the spike reset. Okay, so and it's easy to compute the derivative of this guy, which is the next thing we need to compute in the chain rule, and basically gives you this term. And one thing I dropped here, and I have not talked about, is that I actually also ignored this term. Since we're already approximating, let's just ignore this term also. And you can, if you're worried about it, we can talk later about it. But if you ignore all of these things, you approximate things away, you end up with a learning rule that's actually a 3-factor learning rule, which has many interesting components. So this is ultimately the learning rule for the weight Wij between the presynaptic neuron Sj and the postsynaptic neuron Ui, or the postsynaptic membrane potential Ui. And what do we have here, right? So let's unpack this. So first of all, there's this term here, which is a presynaptic term times some postsynaptic term. And that's in a way, in flavor already like a Hebbian learning rule, which you've heard about earlier. And moreover, you have a nonlinearity here. It's actually a voltage nonlinearity, which comes from our surrogate gradient approach. This function looks like this. It's a monotonically increasing function. It's positive, monotonically increasing to the firing threshold in the membrane potential. That's something we kind of know real neurons also have, because we know you need to depolarize them to induce plasticity. They have voltage-gated calcium channels, they have NMDA channels, so in principle they can implement this, and they might even have it. Finally, there's this, the presynaptic term needs to be filtered, so that's basically the presence of neurotransmitter at the synapse. Then this lambda here from the von Rossum distance implements something like an eligibility trace that you've also heard about earlier, so it kind of keeps a short-term memory of a coincidence. And then finally, there's this ominous feedback term, and that's actually the most interesting one, because there it's the least clear of how neurons implement this. And it really takes care of spatial credit assignment. And there's a lot of ongoing work on figuring out how, in a biologically plausible way, real neural networks could solve this. One thing I should point out, since you'll also be hearing a talk by Wolfram Schulz later this week, is that classically there were these kind of 3-factor rules existed a lot, where this is often in many models the reward prediction error. But there's one important difference here in our problem, is that this should have an index I, and that's actually an insight, I would say, that not all problems are solvable very efficiently if this is a low-dimensional signal that is broadcasted to all neurons. But often it's advantageous, and that really comes out of the math, if there's heterogeneity in this feedback. So each neuron I gets a different error feedback signal. That's something to keep in mind. And it's not entirely clear how the brain would do this. It's not entirely clear how specific it needs to be. And that's an interesting problem to think about. Good. So this was the 2-layer case. It's still nice and easy. We can now keep going, and the math gets more complicated. And as soon as you add a hidden layer now, math gets more complicated, but also you're losing this locality that we just had in the learning rule. So that's an issue. Because now, if you're thinking about this neuron and this plasticity at this connection here, what this depends on here and here is the outgoing weights of these neurons, these guys here, and the activity of these downstream neurons, right? And that's really something that it's very hard to imagine how this synapse can know, and that's something that Crick criticized also in the article. It's called the weight symmetry problem. So there are multiple problems with this. This is a non-local learning rule now. It's not something we want. If you just care about building a spiking network that does stuff, you go ahead with this, and then you have a network that does stuff. If you care now about biology and how the real brain could do it, you have to add further approximations. And there's been a flurry of papers dealing basically with this term. And one of it you've heard, Rafal, I think this is a different summer school. There was another summer school last week, so I get confused. Anyway, so you haven't heard about this, but check these papers. But this all started out with Tim Lillicrap, basically, who realized, we can actually, these weights, you don't need to have them. You could put a random matrix here that you keep fixed, and it still works. So the weight symmetry that you need to propagate information symmetrically from here to here is actually, you can loosen it substantially and the algorithm still works. You always lose a little bit, but maybe not much. And there's a bunch of follow-up papers, and I won't go into much detail here. And the other thing is, of course, the downstream activities. And there's actually a trick that's been used in machine learning. It's called the straight-through estimator, which basically is a fancy name for saying we're going to set this to 1. But if you do this, then, of course, all these interdependencies go away and all your problems go away. And you have a learning rule which looks like this, where the output neuron, let's say, has this membrane potential. These are the spikes you want, but this is the spike you get. This is this error signal coming from the von Rossum distance, here visualized as a function in time. So more, spike less, spike more, spike less is basically what it says. If we look at the input end here, there are a bunch of spike trains. We're just focusing on one connection, this one here. It spikes twice. Then we filter this, because that's the quantity we need here. So that's the presynaptic, this is this X, presynaptic spikes filtered. Then we need to look at the U. The U in this case looks like this. So that's the membrane potential of this neuron here. It spikes sometimes. What we really need is this nonlinear function of the U from the surrogate derivative. This is what I'm plotting here. So it looks similar, but different. Also sometimes when you get close to threshold, it gets big, basically. That's what you see here. And then we need to multiply this guy with this guy. So this with this, no, sorry, this with this gives this, and filtering again. So that's what this means. Complicated, but you have to look at it again to really parse it. But basically, this is now the eligibility trace that detects the coincidence between this being big and this being big, right? And then it's filtered. And here you get this similarly. And now only when these two things together, this whole term, which we just computed, and this term, they're together non-zero, does the weight change. That's the weight change, right? And this happens basically here, but not here, right? And this is the weight change for this weight. So here, this weight should change. Right? So this is really literally how you compute it. It's online. A real neuron could do it. And what you can do with it is, for instance, in this network, you can learn arbitrary input-output mapping. So here you have some input spikes. These are Poisson spikes that's randomly generated, and they're looped, which is hard to see, but they repeat. And there are some hidden layer spikes and no output spikes. And now we just do online learning, and you can see that as we're learning, these representations are refined, and you can literally learn any output spike train you want, so any kind of function approximation you want with this approach. And importantly, to do this, the hidden layer representations also have to change. Right? And that's really at the heart of what deep learning really has optimized in very big, at scale, very big network models, that you don't have to specify anymore what do these intermediate neurons do to solve the downstream problem at the network level. Right. Okay, so this also works for classification. And yeah, I think I skipped this. The only thing that's important to remember is that this randomized feedback can actually learn the problem, whereas if you give all neurons the same feedback signal, right, so like one broadcast neuromodulator, then this is not always good, because then often actually even a simple problem like this cannot be solved. So there should be some sophistication in the type of feedback these neurons receive, and microcircuits presumably do this in the brain, and we still need to figure out how. Okay, so you can scale this up to big networks with excitation, inhibition, speech, and so on, but yeah, that's just details. Good. So to summarize this part, and then I have 10 minutes left for the other part. So the surrogate gradients, they're really approximations that make this optimization possible in spiking neural networks. And if you add further approximations with some kind of hindsight, then you can make these learning rules also look like plausible. And often outcomes with the stuff that doesn't look plausible, outcome in a way predictions or things that you want to think about. How could biology implement this? So that's the top-down constraint that feeds into then looking specifically for something in the neurobiology. And yeah, this effective spatial credit assignment that's linked to this error term remains a topic of intense research, both theoretical and also experimental. But so far we looked really only at these supervised learning problems, right? So meaning that the loss function depends on some kind of output z, before it was s, but here it's now z, sorry. So it's the output and some kind of target, okay? So that's implausible for the brain, because it literally would mean that somebody needs to tell you your hippocampal activity should be this when you're seeing that. That's That's probably not how we work. So what we probably do in animals is a lot of unsupervised learning, where the loss should only really depend on the data, or it should perhaps depend on some kind of temporal sequences of the data. And that's really something that we often also refer to as self-supervised learning, because, for instance, if you think of prediction, you could be trying to predict what's the next input going to be from the previous input. So that's only working on inputs, but it can be very powerful. And in the machine learning nomenclature, it's called self-supervised learning for total confusion, although it is technically an unsupervised learning algorithm. So, and we're going to walk through an example through ongoing work in the lab, which is really on this unsupervised learning, and we'll think about invariant object recognition here as an example. I think everybody here will see that there are cats and dogs on these images. And you don't have any problem doing this, but for machine learning, this was a huge problem for a very long time, solving these problems. So the big question is, how do we do this so easily? And the idea is that there are processing systems in the brain which are essentially similar to deep neural networks. I mean, they're definitely also neural networks, and they're definitely also deep, but they're not just the boring CNNs that we use in computer science. But there's a deep network that takes in inputs that are highly entangled, potentially in the input space, where the input space is defined by the firing rates of the neurons on your retina, essentially the ganglion cells. And they're highly entangled, such that images of dogs lie on some warped manifold, images of cats lie on some other manifold, images of something else lies yet on another manifold. And the only reason why we can differentiate between these is because these manifolds don't overlap completely, so they're separate. But they're usually not linearly separable. It means a single neuron cannot solve this problem. So the ventral system, or the ventral stream in our brains, has evolved to fix this problem. And if it was randomly initialized, it would just map different inputs to different random locations in some kind of output space. But somehow there's plasticity, presumably, that solves this issue and allows us to simply separate now invariant object or recognize invariantly different objects or things or concepts like cat and dog. Right. And so what could be the underlying algorithm? So thinking about loss functions now, what could be an objective that we could design that does this? And one thing that you can think about is, well, when I'm looking at a dog that's running around, of course I'm seeing first the dog running like this, then I get different images as the animal moves, as I move, as the world moves around it. You get different impressions of the same thing. So there's some object permanence over time. And the same thing, if you later maybe see a cat, you would have something similar. Maybe time could be really the teacher here about something that's invariant. And maybe there should be an objective that tries to pull together representations of stuff that co-occur in time. Right? So it just pulls them together. And we're not telling you how it's done now, we just think about the objective. Later we just apply our crank of the learning rule of the derivative, and then we're going to get it. So the problem with this, if you pull everything together, it leads to a representational collapse, typically, which means everything is pulled into one point. Because then you're perfectly predicting everything, right? So if everything is 0, or if everything is a cat dog, right, then you're always right. Like, it's always a cat dog. So nothing wrong with this. But of course, it's completely useless as a representation to disentangle these concepts. So what are you doing? So you need a force that pushes away these representations that don't appear together in time. Right? And so here's an image sequence of a dog, for instance, and you get different images, right? So and they're consecutive in time, as you can see. But now in machine learning, this push force is provided by saying, okay, I'm adding a term to my cost function, which now should push away these things, random images that have nothing to do with what I'm seeing, and they correspond to different objects. That's called a negative sample. And the problem with this is, of course, that usually this is not how we experience the world, right? So it's not that you're all of a sudden in a different film, unless you take the blue pill, then maybe. But otherwise, normally you just see the world like this, right? It doesn't completely switch crazily. So you don't get these negative samples if you're a real brain. So you only get these samples. And of course, sometimes there's a switch, but it takes time. So that's not a viable, plausible solution. So what's another plausible solution? A plausible solution could be Hebbian plasticity. So just as a quick reminder, I'm getting my 5-minute mark, I think. So as a quick reminder, Hebb's postulate was that cells that fire together wire together. And that actually amounts to a local plasticity rule, and it's been found in various settings in the brain. Now, what's also cool is from a computational neuroscience perspective, we know that we can derive Hebbian plasticity rules from an objective which is just maximizing variance. Yeah? So if you maximize variance, you get, for instance, a Hebbian rule like this, where, let's say, all the green points here, like this is already solved, but if all the green points here fall into this region, this is the sign of the weight change, essentially, that's flipped. This is the output activity of the neuron. So these neurons being active, or no, sorry, these inputs being active with this neuron and with the presynaptic activity would increase their firing rate because all the weights would increase. Here, for all of these neurons, the weights would decrease. So that's actually a typical shape for what's called the Bienenstock-Cooper-Munro plasticity rule. And together with my student Manu, we kind of realized, okay, so Hebbian plasticity does maximize variance, so maybe it can provide this push term. And then 5 minutes to go, and then no questions, and then we're perfectly on time. So, so this is cutting it incredibly short. I know this might be a lot of nomenclature overburden, but basically we design a loss function which depends on a predictive term where we say nearby points in time, they should be as close together as possible in this representation space. And at the same time, we want to maximize the variance. And for technical reasons, there needs to be a log. And maximizing the variance is basically minimizing the negative variance. So that's another technicality here. For technical reasons, there needs to be a log. And also for network models, there will be a decorrelation, but I'll spare you this now, that these are details, but you want to prevent neurons from doing all the same thing. Good. So if we plug this together, if we derive the learning rule just by taking the derivative, we end up with something that awfully looks like what's called BCM plasticity, which I've introduced in the previous slide. So here's the weight change, this is the postsynaptic activity. These are different mean activity levels, so you get a sliding threshold, how it's called in the BCM theory. But it's not BCM theory because the theory extends it. So it's compatible with this idea, but it extends it because there's another sliding threshold mechanism that depends on the rate of change. And there's another mechanism, but there's new stuff in this math. Okay? And the cool thing about the BCM resemblance is that this is actually something that seems to be going on in the brain. So we know that in hippocampus or cortex, we find these type of plasticity curves. I mean, not we, but the field as a whole, they have found it. So that's really reassuring. So this could be very close. So if we put this learning rule into a network, so this is what Manu did. So we call this, by the way, LPL stands for latent predictive learning, but it's also local. So if he puts this in a deep neural network, and then he assesses the quality of representations in this deep neural network at each layer, so there are actually 8 layers more than shown here, and he assesses the quality with a linear classifier. So he takes the output of this network at this layer, trains a linear classifier on a big data set with cats and dogs and some other images, and then asks how well does it classify. And you can see that as you add layers, this gets better and better with this learning rule. If you remove this predictive component in the learning rule and you're back to BCM or standard Hebbian learning rule, this goes a little bit up and then goes down. So you're losing a lot. And if you remove the Hebbian component, then you're at chance level. So this is cool because the previous models were down here. The new model is here. It's very similar to the previous, but it adds this little extra, which is important, and therefore a prediction that we're making. And it ties this nicely to this plasticity world. One more experiment. So this was showing you it's consistent with what we know about plasticity. It learns something useful in a deep neural network in terms of representation. So it uncrumples the manifolds by just looking at visual experience. This is now one more experiment I quickly want to show you, done in the DiCarlo lab, actually in monkeys that were freely viewing a screen, and then objects would appear somewhere on the screen. And then naturally, like all primates, also monkeys and us, you saccade to where the object appears automatically, which of course puts a different picture on your retina, right? So the consecutive images that your visual cortex receive, they change. And then they did something they call a swap exposure, where they swap out the object during the saccade. So that's really nifty manipulation. That amounts a little bit to this dog becoming a caterpillar all of a sudden. And if you do this enough times, then actually the selectivity of neurons in the inferior temporal cortex, so the output area of the ventral stream, changes. That's what's shown here. But it's specific to the object and specific to the location, and other controls don't do this. And when Manu does this in our model, the same thing happens. Right. So last but not least, this also works in a spiking network. These are different input streams, Poisson modulated with different firing rates. These, you can see probably that there's something slow going on here. This is the network. These are the inhibitory neurons. After learning, the network activity looks like this. If you try to reconstruct the input from this activity, you see that you can basically only reconstruct the slow inputs, because that's what the network chooses to represent in this through learning. And you can also see that the weights become selective to these slow signals and not the other ones. And if you turn the predictive term off, this is completely lost. And then finally, if you take the same model and you run an STDP experiment on it, the same model will give you an STDP curve consistent with data without being fitted to it. It also describes rate dependence, where this is the pairing frequency in an STDP experiment, the weight change. It also gives you that, again, not fitted to it. And it makes interesting predictions about how the STDP window should change as a function of the history of postsynaptic activity. And you've already seen in Osra's talk that there are many forms of the STDP window, so this is a major big question why they might be there. And here we have a theory why this might be there. I know, sorry, 30 seconds. So I've shown you circuit gradients are an effective tool for building functional spiking neural network and for studying plasticity mechanisms, which is what we're doing in my group. I've shown you several pieces of indirect evidence that the brain leverages this type of prediction, unsupervised learning of future inputs for representation learning. Please try circuit gradients at home if you want. We have some tutorials here. And thanks to my collaborators and my group. And yeah, so we have a PhD program and we're hiring.
Thank you very much for the very interesting talk. You are in well time. We will take 1 or 2 questions. So,
yes. Hello, thank you very much for the presentation. I would like to ask about this common feedback. You mentioned that it doesn't work to reducing error writing, for reducing error. And I'm going to ask, have you tried to use some kind of common noise? Because if you apply common noise in those kind of coupled networks, we increase the synchronization or their connectivities. And after that, have you tried some kind of individualized error or feedback, and did you try if it's work or not? Thank you very much.
Yeah, so to the first question, no, we haven't. And I would like to know a little bit more why you suggest that. Sounds interesting, but we haven't tried it. Second question, yes, we did. And so if we give this more specific feedback, that's actually the Matterhorn spike train requires more specific feedback already. So most problems will require a more specific feedback unless you have a very low dimensional prediction. So for reward, for instance, I think a low dimensional feedback signal works because the reward is just perhaps a one-dimensional signal, and then a reward prediction error would also just be one-dimensional, and I think then this should work with the low-dimensional. But if you're trying to learn a higher dimensional mapping, like, I don't know, muscle outputs, which is like 20, 30 dimensional, it would be very hard to do it with a one-dimensional feedback signal.